This presentation is protected by the US and international copyright laws. Reproduction of the presentation materials without obtaining written permission from the author and Albert Einstein College of Medicine is prohibited.
Today, as the title indicates, I am going to talk about the model we developed for distinguishing lifetime survival, purely based on the genomic profile of individuals. In developing our model, we posit a polygenic architecture for exceptional longevity, which I am going to discuss a little bit later on. Therefore, I start with a brief introduction to the premises of our work and introduce the specific aims and rationales behind them. Next, I am going to explain the framework we used for identifying genes involved in exceptional longevity, and proceed to nuances of our model construction, showing some examples of model performance in distinguishing survival in different populations. I will finish with interesting results from the deployment of our model for identifying proteomic correlates of longevity which can be targeted for drug development. So here is the snapshot of our analysis. We started with tracking allele frequency changes across different age bins, a method we colloquially named it sliding regression. From there, we derived loci with significant frequency change. Next, we tested the association of our shortlisted variants with 614 disease endpoints in the UK Biobank. Our shortlisted variants were significantly associated with 53 traits. We derived PRS scores of these 53 traits across all unrelated individuals in our cohort. We also computed PRS scores for an additional 34 blood and urine biomarker traits. We applied an ensemble of classical machine learning methods to build composite PRS scores and explored the utility of our integrated PRS scores in distinguishing differential survival. Finally, through proteomic analysis we explored the utility of our polygenic model for drug repurposing applications. The premise of our approach relies on 4 key principles. Some of these assertions are better supported than others, but together they provide a plausible assumption for the construction of a predictive genetic model for longevity. First and foremost, we posit that a large number of loci influence variation in lifespan. I am aware that some are of the opinion that longevity is a rare condition, hence there should be a handful of rare variants with large effect sizes underlying exceptional longevity. It comes as no surprise that whole exome sequencing experiments, so far, have fallen short of identifying a slam-dunk variant, and more or less, they rapidly turned into a fishing expedition. We also posit that the allelic effects on human longevity are well approximated by a simple additive model. Therefore, polygenic scores should, in principle, account for a substantial proportion of the phenotypic variance. Third, with a potentially infinite number of measurable traits and finite genetic variation, the genetic basis of traits would inevitably overlap. Since aging is the ultimate outcome of multisystem functional decline, pervasive pleiotropy is inevitable. For example, variants that delay the age of menic in women tend to delay the age of voice drop in men, decrease body mass index and increase adult height. And finally, due to the pervasive pleiotropy, the human lifespan is indirectly under some sort of stabilizing selection toward an optimum. So, we formulated 2 specific objectives, each justified with a rationale. Our aim 1 was to identify genetic variants that affect survival to a given age. As discussed earlier, the idea is that common genetic variants underlie much of the variation in lifespan. The rationale for aim 1 is the fact that longevity is heritable. As a matter of fact, narrow-sense heritability is estimated in the range of 10 to 20%. Our second objective is to construct a predictive model for human longevity based on the additive effect of alleles. The underlying rationale is the fact that polygenic scores should, in principle, account for a substantial proportion of the phenotypic variance. We adopted a method to examine whether individual genetic variants or sets of genetic variants currently influence viability. In this framework, colloquially named it sliding regression, we track allele frequency changes in consecutive overlapping sub-gene pools that are sorted based on definitive age at death of participants in the Einstein longevity cohort. The idea behind our approach is that, if a genetic variant does not influence viability, its frequency should remain relatively constant at all ages. Whereas variants with a negative impact on longevity are expected to be naturally purged from the centenarian gene pool. And ones with a positive contribution to healthy aging are expected to be enriched in the gene pools toward the extremities of the human lifespan. By fitting a regression model where the outcome is that the sex-corrected frequency of the alternative allele within the age bin, beta is the slope of regression, E is the age at death, and alpha is the intercept of regression defined by the expected mean of the alternative allele frequency in our discovery cohort, we would be able to quantify the effect size of each allele on survivability selection. This approach is similar to GWAS for longevity, but it does not focus on the endpoint, rather, it quantifies any shift in allele frequencies with age. Overall, we tested about 8 million variants, for which 8.2% of them reached nominal significance, and only 22 variants surpassed the genome-wide significance threshold. The x-axis in this volcano plot represents the slope of regression, with prolongevity variants that their frequency increases by age, are clustered to the right of the plot, and proaging variants with negative slopes to the left of the plot. The y-axis shows the p-values in logarithmic scales with genome-wide significant variants identified in red. As with many GWAS analyses, the majority of these genetic variants are turning up in non-coding regions of the genome. Interestingly, 14 of these variants were revealed to be EQTL loci. At the bottom you see the trends for 3 ApoE haplotypes. As expected, ApoE4 haplotype shows a decreasing frequency toward the extremities of lifespan. When we mapped the variants to the nearest genes, only lipoprotein A and LDL receptor genes were among the genes previously reported to be involved in aging. We know that exposure to high lipoprotein A levels is associated with coronary heart disease, and it is recently shown to be causally related to shortened parental lifespan in the UK Biobank. We, therefore, supplemented positional mapping with EQTL and chromatin interaction mapping using the GTEx and PsycEncode data. We identified a relevant gene with plausible functional implications in longevity for each risk locus. I only show 2 examples of the most interesting cases for the benefit of time. The most prolific regulatory activity was identified for a risk locus on chromosome 9. This risk locus is about 86 kilobase, and 34 genes are prioritized by both EQTL mapping and chromatin interactions. Here in this circus plot, the blue segment shows the physical position of the risk locus in the chromosome. Genes prioritized based on EQTL mapping are colored green, and genes prioritized based on chromatin interactions are shown in orange. One of the interesting genes prioritized in this locus was monosidase, which may explain the link between type 2 diabetes and Alzheimer's disease. Monosidase 1 is involved in the intracellular clearance of misfolded alpha-1 antitrypsin. Analysis of post-mortem histopathological samples from AD patients has shown that accumulation of misfolded alpha-1 antitrypsin plaques occurs in the lesions of AD patients. It is also shown that properly folded protein protects against amyloid-induced toxicity in microglial cells. Interestingly, treatment of diabetic mouse models with properly folded alpha-1 antitrypsin resolve glucose intolerance and normalizes blood glucose level. Due to the decline in autophagy during aging, the concentration of misfolded alpha-1 antitrypsin increases. Given the scavenging role of MAN1B1 in clearing misfolded proteins, its elevated expression during aging renders a protective effect against misfolded alpha-1 antitrypsin. Here at the right bottom corner is the evidence from a relatively recent study showing the effect of alpha-1 antitrypsin treatment in reducing amyloid severity. When pancreatic islets were cultured in the presence of alpha-1 antitrypsin, amyloid severity was reduced 6-fold after 2 days in culture. Even after 7 days of culture, when amyloid severity was very high, alpha-1 antitrypsin reduced these levels more than 2-fold. Another exciting example was observed on chromosome 2, where 2 genes were prioritized by EQTL mapping. These 2 genes have been recently shown to be functionally relevant in the context of aging. Suppression of GLS in aged mice is shown to alleviate aging-related phenotypes through the elimination of the senescent cells. As shown in the bottom, mice treated with BPTES, an inhibitor of GLS, had a lower number of atherosclerotic plaques, a lower number of lesions, reduced adipose weight and improved hanging endurance and grip strength compared to aged mice, perhaps hinting at a protection mechanism against frailty. Similarly, inhibition of GPD2 is shown to further extend the lifespan of DAF2 mutants in C. elegans. Here, in this Kaplan-Meier survival curve, reduced mortality of DAF2 mutants, which is shown in blue, is comparable with control and related treatments. Given that suppression of both genes is identified to be associated with a longer lifespan, we believe the risk locus we identified in our centenarian cohort exerts its influence through the downregulation of both genes. Next, we carried out a phenome-wide association analysis to explore the pleiotropic landscape of the prioritized variants. PheWAS are an unbiased approach to test for associations between a specific genetic variant and a wide range of phenotypes across large numbers of individuals. Overall, we tested the association of 1,504 traits and disease endpoints across 28 domains in the UK Biobank, of which 178 traits and diseases across 17 domains were identified to be significantly associated with the candidate variants. PheWAS associations were significantly enriched in 3 domains: skeletal domain, that included traits such as bone mineral density; metabolic domain, that included traits such as body fat percentage, and overall cognitive performance. This brings us to aim 2 of our analysis. Multimorbidity is a common phenomenon during aging. The shared genetic component among age-related traits and age-related diseases results in cross-trait pleiotropy and underlies certain multimorbidities. Given the ubiquitous pleiotropy revealed in the PheWAS analysis, we constructed a model that integrates the complex interplay of pleiotropic aging-related traits in shaping longevity. We applied a stacked ensemble method to construct the integrated longevity genetic score. We first randomly split the Einstein centenarian cohort into the derivation set of 715 individuals and the validation set comprising 273 individuals. We then used polygenic risk scores of 53 select PheWAS-identified traits and 34 additional UK Biobank blood and urine biomarker traits. We trained the ensemble model on 65% of the derivation dataset and tested it on the remaining 35%. Upon 5-fold cross-validation, our model achieved an area under the curve of 87%. The final stacked elastic net regression framework shrank coefficients of 10 traits to 0, and the final score was constructed using the coefficients of the remaining 72 traits and 297 interaction terms. The heat map plot is showing the interaction between the traits' polygenic scores used in constructing the iLGS. Interacting pairs with positive coefficients are shown in red, and pairs with negative coefficients in blue. The numbers inside the heat map boxes correspond to the size of interacting pairs coefficients in the final model. The bar plot to the right shows the number of interacting pairs per trait polygenic score, which are colored based on their domain category. The highest absolute number of interactions are mediated through the insulin-like growth factor 1 with 17 interacting partners, which is followed by the rheumatoid factor with 15 interacting partners, and alkaline phosphatase, and estradiol, both with 14 interacting partners. Among the remaining domains, traits within the cognitive function class have the second highest number of interactions with other traits. In this domain, educational attainment interacts with 14 other traits, representing the most important non-biomarker traits in describing the additive variant in our model. When traits were considered individually, age at first birth, anxiety, and walking pace have the most interacting partners, following educational attainment. Using the validation portion of our in-house centenarian cohort, we applied a multivariate Cox proportional hazard model to evaluate the association of iLGS with age at death while controlling for sex, ApoE4 status, and their interaction with iLGS. In this forest plot, since we are investigating the association of each variable with age at death, those increasing the chance of mortality align to the right of the plot, and those reducing the hazard for mortality are to the left of the plot, expectedly below the hazard ratio of 1. Here we show that iLGS, which is a composite polygenic quantity derived from centenarians, is significantly associated with age at death in the validation cohort. To put it into the context, for every 1 unit increase in iLGS value we see 3% decrease in the baseline mortality hazard. We also investigated whether the association of iLGS is mediated through gender status of ApoE4 haplotype, for which no statistical interaction was identified. This shows that the association of iLGS with survival is independent of gender and ApoE4 status. We also carried out a stratified Kaplan-Meier analysis across the quintiles of iLGS to investigate if survival curves are significantly different across the top and bottom quartiles compared to the interquintile range. As evident here, iLGS is highly powered to distinguish differential survival at the quintile level. Comparison of iLGS at decile level shows, individuals in the top decile live up to 4.8 years longer. Next, we sought to replicate the predictive performance of iLGS in two separate cohorts specifically ascertained for healthy aging. The first cohort is the elderly cohort of the Scripps Institute, comprised of 511 healthy elderly between 80 to 105 without any chronic conditions or medication taking history. The second cohort is the Medical Genome Reference Bank of Australia, comprised of 2,500 individuals who lived at least 70 years of age without any history of cancer, dementia, and cardiovascular disorders. In both cohorts, we investigated the association of top and bottom quintile iLGS with three age categories. The forest plots illustrate the enrichment of top and bottom quintile iLGS scores across three age bins. Since the absolute age at death for participants of these two cohorts are not available, we formulated the null hypothesis as no association between the top and bottom quintiles with age category at the last point of contact, and tested for evidence against the null using a Fisher's exact test. In the elderly cohort, the top quintile was significantly associated with both the 95+ and 90 to 94 age categories. Individuals in the top quintile are 3.67 times more likely to surpass 95 years of age and 2.49 times more likely to fall into the 90 to 94 age bin than the rest of the iLGS strata. In both replication cohorts, the strength of association between the top quintile and the older age categories is significantly stronger. Given that age at death in these cohorts is undetermined, we believe the stronger signal toward exceptional longevity arises from the more homogeneous cohort construct among individuals achieving the upper limits of longevity. Using the available clinical data in the MRGB cohort, we further investigated the association between the top quintile iLGS and three age-related traits, including extreme obesity, identified by BMI over 40, treatment history for high blood pressure and treatment history for high cholesterol. Despite the expected direction of effect across the three traits, the protective effect of top quintile iLGS is only statistically significant against treatment for high blood pressure. The quantitative size of iLGS is determined at birth and remains constant over an individual's life course. Given the association of iLGS with longevity, we searched for the proteomic correlates of iLGS among the 237 individuals in the validation cohort. We identified 40 proteins, of which 37 are positively associated with iLGS and 3 are negatively associated. Previous studies have repeatedly replicated the association of pleiotropin with both chronological age and extended lifespan. Also, the association between the expression of SEMA4B and the favorable proteomic signature of intermittent fasting has been previously reported in the context of aging studies. The connection of MAT2B to aging has not been studied, although its overexpression has been attributed to poor prognosis of triple-negative breast cancer and critical illness in COVID-19. We queried the drug bank database to identify drugs targeting or interacting with the 40 iLGS-associated proteins. Our rationale was that the underlying mechanism of action for drugs interacting with iLGS-associated proteins might implicate the pathways relevant to longevity and they could be repurposed as anti-aging treatments. One drug, fostamatinib, interacting with at least 4 longevity-promoting proteins, is a selective inhibitor of spleen tyrosine kinase that acts upstream of the PI3K ACT pathway, which is upregulated during aging and whose downregulation promotes improved cell survival in neurons. The suppression of SYK attenuates the PI3K ACT signaling, which inhibits mTOR and activates FOXO, leading to the activation of propapoptotic processes and the cell cycle arrest. Although its precise mechanism of action is unclear, the active metabolite in fostamatinib has been recently found as a novel senolytic agent. Given the extent of its interaction with longevity-promoting proteins, this drug merits further investigation as a potential anti-aging treatment. So in summary, as discussed, our analysis constituted 6 major steps. In step 1, we used the sliding regression framework to track allele frequency changes across age bins in the Einstein centenarian cohort. We fitted a robust linear regression with estimator for each variant to derive the slope and p-value of association with chronological age. In step 2, the association of candidate variants with 1,504 disease endpoints and traits were investigated in a PheWAS analysis. For each variant, we used the Bonferroni-corrected association p-values for shortlisting associated traits. In step 3, we used GWAS summary statistics across the 87 traits, primarily from the UK Biobank, to calculate the polygenic scores for all participants in the Einstein centenarian cohort. In step 4, we split our cohort into the derivation set of 715 individuals and validation set comprising 273 individuals. Using the derivation portion of our cohort, we applied a stacked elastic net regression framework to effectively combine polygenic risk scores across the 87 traits and construct a composite prognostic score to distinguish survival chances. We trained the ensemble model on 65% of the derivation set and tested on the remaining 35%. Upon fivefold cross-validation, our model achieved an AUC of 87%. Using coefficients derived from our stacked model, we computed the integrated longevity genetic scores. Subsequently, using an external cohort, we converted the scores to a set of 3.8 million variant weights. In step 5, we carried out survival analysis to test the performance of iLGS in predicting survival in the validation set as well as 2 external cohorts including the Welderly and MRGB cohorts. In step 6, we carried out a gender-stratified association analysis to identify proteomic correlates of iLGS. Proteins significantly associated with iLGS were subsequently queried in the drug bank database to identify druggable targets. Finally, drugs targeting or interacting with iLGS-associated proteins were investigated for potential drug repurposing as senolytics. So, in conclusion, we showed that how pervasive pleiotropy could be leveraged in constructing a composite polygenic score for longevity. Identifying individuals at increased risk of death enables targeted lifestyle modifications earlier in life which result in increased survivability. Unlike most established predictive factors for adverse events, including BMI, blood pressure, etc., that may vary over time and are typically not informative, the polygenic scores are stable and can be derived from birth. In conclusion, application of iLGS to the extant populations offers an opportunity for better risk management among individuals in the lower end of the score distribution.
This presentation is protected by the US and international copyright laws. Reproduction of the presentation materials without obtaining written permission from the author and Albert Einstein College of Medicine is prohibited.